Hi guys, this is Dr. Divya Pradhan, and today we are going to talk about animals and how they evolved, particularly the invertebrates. Alright, what are the distinctive characteristics of animals? First and foremost, animals are multicellular and they are eukaryotes. Okay, so they are multicellular. That means obviously you understand this, that they have many cells. They are eukaryotes, meaning to say that they have a distinct nuclear membrane. That is what is the meaning of the word eukaryote. Surely you know that. They are heterotrophs. That is, they acquire nutrients from external sources, carry on sexual reproduction, have specialized tissues and organs. Understand what tissues are. Tissues are special cells doing special function. Okay? And these tissues are organized into organs, and of course they have muscles and nerves which allow the animals to perform flexible movements, search for food, seek mate, shelter, suitable climate, etc. Well, regarding the origin of animals, the scientists have put together a colonial flagellate hypothesis. So what this says is that the animals descended from an ancestor that resembled, right here, a colony, just a group of flagellated cells. These flagellated cells then organized themselves into hollow sphere, as you can see. Some cells became specialized, so of course you had tissues, and they started doing special functions, and then finally this hollow ball of cells invaginated, and so this is how germ layers, body layers developed, and so you had an outer layer and an inner layer, and finally, of course, then you developed a third layer later on. And of course this implies that the radial symmetry, which we'll talk about in a few minutes, came before bilateral symmetry. We'll talk about this in a few seconds, okay? So here is the phylogenetic tree of animals, and this is based on molecular. Obviously, for molecular we are going to be using DNA, and anatomical, comparative. We are going to compare the anatomy, and then they drew up this phylogenetic tree. So of course you need to understand that although we do not have, right here, although we do not have any particular, one second, we do not have any fossil record by which to trace, but this tree is mostly based on, particularly on DNA, more than anatomical. All right, we will use anatomy. So here, the first anatomical feature that we are using is symmetry. Well, is it asymmetrical, radial, or bilateral? A means that there is no symmetry, no body shape. Look at these sponges, they have no body shape. Radial symmetry is that you can cut like the spokes of a wheel. This is arranged circularly, like the cnidarians. Now, for example, Hydra. So you can cut along any of these of the diameter, and you will get two equal halves. Bilateral symmetry means that there's distinct right and left part of the body. So this, of course, is accompanied by cephalization. Cephalization means formation of the head. Cephala means head. Some people call it cephala, some people call it cephalization. So this is the bilateral, the asymmetrical, radial, and bilateral symmetry. The second anatomical feature that they will compare are the germ layers. How many body layers? So the body layers, you can have either no body layer, for example the sponges, or you can have two germ layers, for example the cnidarians, and then you can finally have the three germ layers, the ectoderm, mesoderm, and the endoderm, as we can see in flatworms. All right, we'll talk about this in a few seconds. So as per looking at their evolution and development, the scientists had divided animals into two groups, protostomes and deuterostomes, depending upon cleavage, phytoblastopore, and coelom. Let's say cleavage. If the cleavage is spiral, they are protostomes. They belong to the lower forms. Deuterostomes are the higher forms of invertebrates, more evolved species. Cleavage is radial. Well, what about phytoblastopore? If the blastopore becomes the mouth, then they are protostomes. If the blastopore becomes the anus, they are deuterostomes. Well, what about the development of coelom? First of all protostomes do not have coelom. Coelom is found only in some protostomes. But of those, the coelom develops by the splitting of the middle layer, that's the mesoderm. Okay? Whereas in the deuterostomes, the coelom develops by the outpocketing from the primitive gut. Moreover, the protostomes are subdivided into two. And so subdivided two groups are the ecdysozoa, that is, they have molting, and the trochozoa. So here the ecdysozoa, they both are the roundworms and the arthropods. Trochozoa, they have a trochophore larva, either now, presently, or they have had sometime this larva in their development. So the first phylum that we come across are the sponges, phylum Porifera. Porous. Porifera should remind you of the word porous. So here, these are the perforated forms. They have no tissues. They have a cellular level of development. That is, of course, it resembles just a group of flagellated cells that are living together in a colony, very primitive. And of course, they are asymmetrical. We talked about this before. They are filter feeders. What does it mean? They are going to pump in water, and as the water is circulated through the pores into the central body cavity, as you can see here, it is going to trap any food and of course digest the food. Now here, what about the skeleton? These group of cells are supported by spine-like needles, and these are called spicules. Okay, and these are either single-rayed or three-rayed or six-rayed, and of course reproduction is mostly asexual reproduction. That means they do not require a partner. But sometimes they can have sexual reproduction. Asexual reproduction is mostly by budding. Sexual reproduction, they form sperm and the egg. But fertilization is outside. The special cells are released outside. Fertilization is outside the body. So here, as you can see, remember I told you that there are a group of ciliated cells. These ciliated cells are called the choanocytes, collar cells. These are the ones which have the collar and the flagellum. They will pump water, so water is drawn in through the pores into a central cavity and released outside through the osculum. All right, nothing very fancy. The next phylum are the cnidarians. The cnidarians are radially symmetrical. So here, first and foremost, so here's one step up. They are radially symmetrical. What about second thing? Here they have a tissue layer of organization. So the cells have formed special tissues, but no organs, no system. And second of all, or third of all, they have bilateral symmetry. If you remember what bilateral symmetry is, that they are arranged in a circle, in a round format. So here they have two germ layers, the ectoderm and the endoderm. And of course, here we can see that this is the typical hydra, typical form of the cnidarian. There are two body forms, as a polyp where the tentacles are up, or the medusa. I think you have seen this jellyfish, Finding Nemo, etc. So the tentacles are down. Now these tentacles, focus on these tentacles because I'll be talking about these tentacles. These tentacles have very special cells. Here you go. These special cells are called cnidocytes. These cnidocytes have a needle-like structure in this cyst, in this fluid-filled bladder called a nematocyst. And as soon as a prey comes and touches this fine needle, then the entire spring-like syringe will spring out, paralyze the prey by injecting it with this paralyzing fluid. And as the paralyzed prey, the tentacles here are going to capture the food, bring it to the mouth, and digestion will take place in the gastrovascular cavity. Of course, the gastrovascular cavity is just a blindly ending sac. All right. Now, here with this, we come to the end of our slide, and we will talk about it in the next slide. Please contact me if you have any problems or issues, and thank you for listening to me.